It is my pleasure to welcome you to tonight's installment of the Silverstein Lecture Series. This series is made possible by a very generous gift donation from Herman and Bea Silverstein to promote understanding of medicine and the advances of genetics that are improving our lives. And they fund 2 seminars a year to bring in internationally acclaimed experts to tell us about new areas of biology that are going on. And tonight we have a very exciting topic. It's going to be discussing how the communities of microbes that have been with us since time began, how they interact with us and how they influence our health. And our expert tonight is Dr. Susan Lynch, who did her undergraduate training at University of College Dublin in Ireland, and actually did her graduate work there as well in microbiology. From University College, she went to Stanford University, and from there in 2005 to the University of California, San Francisco, where she has been a just rising star whose potential is unlimited as far as we can tell. And she has received numerous awards, and she is very well funded, which is something to be noted in this era of challenging funding. And she is going to be our guide tonight as she introduces us to the world of the microbiome. So let's welcome Dr. Lynch.
Thank you so much, Greg, for that really very nice introduction. And thank you everyone for turning out tonight to hear about your microbial self. This is a very new and emerging field, and I'm very excited to share with you some of the work that we've been doing and others in the field that really are starting to make us feel like that this area could be very transformative for human health. And so I feel incredibly fortunate to be a microbiology in the current era. I just want to begin by bringing you through where microbiology has come from. And in the early days, really the genesis was in 1676 when Antoine van Leeuwenhoek identified what he called animalcules in the dental plaque from his mouth. And that was the first observation of microbes that's recorded. We then move on a few hundred years later to Theodore Escherichia, who isolated Escherichia coli from the feces of bottle-fed infants. And given what I'm going to tell you a little later on in the lecture, I think that this was a really interesting observation, that this was isolated from the feces of children who were fed by bottle and not fed by breast. We move on for several hundred years, and we really did most of our work on single species. We looked at pathogenesis, we examined how organisms colonize surfaces, and it's really only in the last 10 years or so, with the advent of new technologies, that we have dramatically shifted our view of human-associated microbes. We had, for the first time, identified that there are far more than the 1 or 2 microbes that we had traditionally studied that were largely pathogenic. We had largely spent most of our efforts studying those organisms that cause disease. And what we started to understand was that there's a whole lot of other organisms out there that colonize the human body. And so how do we know this? Well, we stand on the shoulders of giants in the field of environmental microbial ecology. People like Norm Pace and Alan Konopka, way back in the 80s, realized that the morphology and just the different types of microorganisms that you could observe microscopically in a milliliter of seawater could never be recreated or cultured on a traditional agar plate in the laboratory. And so they set about looking for molecular approaches to identify organisms without having to culture them. And they came up with this gene, the 16S ribosomal RNA gene, as a biomarker for bacteria. It's found exclusively in bacteria, the bacterial kingdom, in all bacteria, not in fungi, not in humans, not in any higher organisms. So it's a wonderful gene for identifying bacteria. And that's supported by the fact that it has these, this is the gene, this is a cartoon of what the gene looks like. It's about 1.5 kilobases in length. And it has these regions of sequence that are highly conserved. They're very similar across all known bacteria. And they flank these, what we call hypervariable regions. There's 9 of them in total. And they evolve at different rates across bacteria. So their sequence is very different across different bacterial species. So what you can do is you can take your sample, be it an environmental sample, a stool sample, an airway secretion sample, extract the DNA from that sample, and use primers, so little pieces of DNA that we use to make copies of a region of a gene, to amplify these hypervariable regions of the gene. And then we basically read the sequence of these hypervariable regions, and that allows us to understand which bacteria they came from in the sample. And so instead of culturing things out, we're getting a profile of all the distinct types of bacteria in a specific sample using this molecular approach. So we get a far higher resolution view of the types of organisms in a given sample. So when we've applied this, which had been applied for a couple of decades to environmental samples, when we've applied this technique to human samples, what we've found is we're not alone. We are overtly colonized by microbes both on our surface, on the skin, and on all the mucosal surfaces, or the soft linings that line the gastrointestinal tract, the respiratory tract, and the urogenital tract. They are overtly colonized by microbes. And it's quite humbling. We have clearly co-evolved with these organisms for eons. 10% of the cells in the human body are human. The other 90% of cells in the humans are microbial. What's even more astounding is we're a single species, and we encode about 22,000 genes. That's 1% of all the functional genes in this superorganism. 99% of the genes that exist in this ecosystem are microbially derived. And so these are not just simple bystanders watching the host pass by through life. These organisms have evolved with us to provide us with key functional capacities that we ourselves do not encode. They can digest indigestible carbohydrates. For example, fermentable fiber is fermented by organisms in the gut to produce short-chain fatty acids. These are essential. They are an energy source for the cells that line the gastrointestinal tract, and they're also potently anti-inflammatory. These organisms produce vitamins and hormones. They are critical for immune development, and we know that because if we take mice that are germ-free, they have no microbes colonizing them, they are both immunologically and physiologically aberrant. They do not develop appropriately. You can only rescue that phenotype if you colonize those mice with commensal bacteria. We and others have also shown that key organisms in the gut microbiome are really necessary for maintaining immune homeostasis, so keeping the host immune response in check. We've shown that an organism called segmented filamentous bacterium, for example, induces an inflammatory response driven by Th17 cells, which is associated with a large number of chronic inflammatory diseases and autoimmune diseases. We've shown this in mice. We've not yet found the parallel in humans. But it shows that specific organisms, when they're enriched in the gut of at least these mice, can drive particular immune responses that we associate with disease. Another thing we've shown in our studies of the sinus microbiome, this is an area that we thought was sterile until a few years ago. And what we've shown is that the microbes on the sinus mucosa can actually protect that surface from pathogen infection. If you deplete the sinus mucosal microbiome and then instill a pathogenic organism, you actually drive a greater degree of pathogenesis compared to if you still instill that pathogenic organism into a mouse that still has an intact, native, healthy microbiome. So these organisms protect our mucosal surfaces in the healthy state. The other thing we've realized is that for every site we've looked at, so being the skin, the external auditory canal, the mouth, the gastrointestinal tract, each one of these sites is its own distinct ecosystem and houses its own specific relative ratio of organisms at each site. And that's shown here. This is phylum level relative ratios of organisms. So, for example, in the gastrointestinal tract, in Western nations at least, there is a 3:1 ratio of Bacteroidetes to Firmicutes. And that's very distinct from what we find in the esophagus or the nostril or at other sites. So that tells us that in a healthy individual, there are distinct ecosystem conditions that select for specific organisms in that site. And so this seems like a whole lot of data to deal with and a whole lot of differences and variability across humans. It is, but we have a framework to understand the human microbiome because we've studied ecology for decades, and we have principles and theories that the human microbiome obeys. For example, we know that we disperse microbes between one another, and that's evident from wonderful work from Jeff Gordon, who has shown that family members share more similar microbiomes in their gut compared to non-family members. We also know that microbiomes can be invaded, much like we see in other ecological systems, and this is frequently associated with infection, where we see invasion of a pathogen into an unstable microbial community. We also know that there is dispersal from mother to child during the birthing process, and this is really key, we think, and I'll come back to it in a few slides, in the foundation for developing the gut microbiome in early life and protecting or perhaps contributing to disease development. And then, like other ecological systems, we undergo this period of what we call primary succession ecologically. We develop a microbiome in a previously pristine environment. So for example, in the gut, we develop a microbiome over the first year of life. And again, I'll talk about this in a later slide. And we are, as I mentioned, particularly interested in this period of life. We're also interested in what goes on in utero, but today I'm going to focus on our interest in this very early stage of life and how it may contribute to health or disease status in childhood and we think beyond, but we're focused today on childhood. And we're interested in that period of life from the perspective of childhood allergic asthma development. This is something that, asthma is something that we can clinically diagnose when children are about 6 years of age and be confident of the diagnosis. It is a disease that has raised in prevalence quite dramatically over the last several decades, although recent data suggests that this has now flattened out, this very alarming curve. And the other interesting point is that it is so prevalent in Western nations compared to developing nations. So there's something about Western lifestyle that really is driving, or is at least contributing, to the childhood allergic asthma epidemic that we're witnessing. And this, I hope you can read, is taken from a really wonderful paper from Graham Roke. And it really just tells us, where have we come from microbiologically as humans? Well, you know, we started off in the Paleolithic age as small group hunter-gatherers, so we had a lot of environmental microbial exposures. And even as we moved through the ages to the pre-industrial era, we still largely lived in rural environments. A lot, again, of microbial exposures. We began to domesticate both animals and cats, dogs, and began agricultural practices where we had cattle in close contact with us, and we had a lot of untreated water consumption. So again, a large exposure to microbes in the environment. In the modern era, post-industrial revolution, we largely live in cities. We are surrounded by concrete, and we have really dramatically changed our interaction with the microbial environment. And this has led to the proposal of the hygiene hypothesis by David Strachan, and it's been revised several times since its original incarnation in the 1980s. But it basically posits that a lack of exposure to microbes, particularly commensal microbes, is what is driving the prevalence of allergic asthma and a number of other chronic inflammatory diseases in Western nations. And so when we think about allergic asthma and allergy, and allergy is a precursor quite frequently to developing asthma in childhood, so we are particularly focused on the development of allergic disease. And epidemiological studies have identified a large number of factors that are related to disease development in childhood. They include early life antimicrobial exposure, formula feeding, cesarean section, lack of maternal exposure to animals during pregnancy, or maternal consumption of antimicrobials during pregnancy. And it turns out that the more rounds of antimicrobials the mother takes during pregnancy, it increases the risk for allergic asthma development in her offspring. Also, the lack of early life furred pet exposure is an associated risk for developing disease. And when I look at these, I just think of factors that impact the gut microbiome. These have all been shown. We know in adults, antimicrobial exposure causes an acute depletion of the microbiome, but it's quite resilient at that stage because it has developed and it's relatively stable, and it bounces back within a few weeks to a composition that largely resembles that before the antimicrobials were administered. However, there are specific organisms that never are found in those communities after antimicrobial exposure. And thinking in terms of the amount of function that these organisms confer upon us, it's possible that specific functions are therefore lost, and if they're critical functions, you can imagine that disease may ensue. Formula feeding reduces the number of commensal bifidobacteria in the infant gut. Caesarean section delivery results in a very interesting and dramatic difference in the types of microorganisms that are found in the infant in the very early days of life. Children who are born by C-section are colonized by Staphylococcus and Streptococcus, species that are found on the skin, and this is because this is their first exposure. Children who are born vaginally are colonized by Sneathia and Lactobacillus species found in high abundance in the mother's vaginal tract. And if we think about our ecological framework, ecologically, founder species frequently dictate the types of organisms that co-colonize in an ecosystem. So again, you can imagine that having this aberrant microbial colonization in early life may lead to the development of an aberrant microbial community that lacks specific functions necessary for immune homeostasis activation. And lack of early life for pet exposure is kind of an interesting one. I'm going to circle back around to that in the latter part of our talk, how Fido is good for our health. But what this tells us is that everything is pointing to early life exposures, either in utero and particularly early postnatal microbial exposures being important in defining host health or childhood allergic disease development. And so this is supported by observations based on culture-based studies by John Penders and subsequently by Kaliamaki and colleagues, showing that if they looked at the types of microbes that were present in the feces of children who were just 3 weeks of age, children who had a lot of Clostridium difficile or Escherichia coli had a significantly increased risk of developing allergic asthma. So this tells me very early life, a dysbiosis or a perturbation to the microbiome is associated with disease development. And why is this period of life so important? Well, remember I told you about primary succession, this ecological phenomenon where a community of species develops in a previously pristine environment. This occurs in humans. This is simply a plot where we've collected stool samples from children at birth, 1, 3, 6, 9, and 12 months of age, and we've profiled their gut microbiome. And we've simply calculated how diverse the communities are. And what you can see is it doesn't matter which children it is, there's this dramatic diversification of the gut microbiome over the first year of life. And this has been shown by David Relman and Ruth Lay and others in very small studies. So the gut microbiome diversifies over the first year of life, and we know we get an inoculum, and it looks like this is the diversity of that inoculum that we receive in the birthing process. So our first question was, where does all the rest of that diversity come from in the human infant gut microbiome? And in Western nations, we spend most of our time indoors. We really are disconnected from the external environment. We spend almost 90% in the built environment, 90% of our time in the built environment, and almost 70% of that in our homes. And so we became really interested in hypothesizing that the microbial environment in the household maybe influences the microbiome, specifically the gut microbiome, which then drives the immune response of the host and develops, contributes to allergic disease development. And we're thinking of this as how the environment may actually this linkage that we know between the environment and disease may be mediated through the microbiome. And so the first question we had was, are early life house dust microbial exposures related to the development of atopy or allergic wheeze in children raised in those houses? Because you could imagine that if children have specific microbial exposures, that they may have a very different gut microbiome and a very, according to this hypothesis, a very different propensity to develop disease. And so we had the wonderful opportunity to work with Jim Gurn and Bill Bussey in the Inner City Asthma Consortium, an NIAID-funded group, to examine the really high prevalence of allergic asthma in inner city populations. And Jim and Bill had a prospective cohort of full-term infants born in low-income inner city urban neighborhoods. There were about 600 families that were enrolled. The sites were Baltimore, Boston, New York, and St. Louis. And they had the foresight to collect house dust during the first year of life from the homes in which these children were raised. And then they defined their allergic status at age 3. And so this was, these were the samples that we worked with. There were 104 dust samples that we knew the allergic outcome of the child at age 3, and we had a parallel first year of life house dust sample available. And we had atopy defined as an IgE, serum IgE level of greater than 0.35 for any allergen, and wheeze defined as at least 2 episodes of wheezing with at least one of those episodes in the 3rd year of life when we're assessing their atopic wheezing status. And we could break our 104 samples into 4 groups. Children who developed neither atopy nor recurrent wheeze, children who were simply recurrent wheezers, those who were only atopic, and the more severe children who developed both atopy and recurrent wheeze. We profiled the house dust microbiome of the homes in which these children were raised. And the first thing we did was we took that vast profile of microorganisms and collapsed it down into a single metric to summarize the community composition. And we used 3 of them. Microbial community richness, which is a measure of how many different types of microbes, or bacteria in this case, are found in the sample. Microbial community evenness, the relative ratios of those organisms in the community, and the diversity of the communities. And you can see, though none of these quite reach significance, that there's this trend towards fewer types of microbial species in the environment of those children who go on to develop the more severe atopy and recurrent wheeze. There are also more uneven communities, suggesting that these children are exposed or overexposed to certain organisms. And then their diversity is also significantly reduced. And I should say that this diversity index is calculated based on these 2 metrics, so we would expect it to be reduced. If we delved a little deeper and start looking at the actual composition of these microbial communities, there emerged an even more interesting picture. These are called non-metric multi-dimensional plots. Don't be scared, it's pretty simple. Each spot represents a profile of microbes in a single community, and its distance to another spot tells you how similar the microbes in this sample are to the microbes in this sample. And so you see how similar, for example, this sample is to all other samples that we profiled. And then we can do some statistical analysis on this and ask how similar or distinct are communities in, for example, children who wheeze versus those who neither wheeze nor are atopic. And essentially, they're not that different. They're pretty similar communities, the house dust communities. Then we ask, what's the difference between atopic and neither, or is there any difference? Yes, there's a significant difference in the types of microbes found in the atopic household versus the households where the children do not develop disease. And this becomes even stronger when we look at the homes where the children develop atopic wheezing versus those who do not. So there's something different about the microbes in those homes. And so we next asked, what is that difference? At a very high level of resolution, who are the specific microbes that discriminate developing disease versus being protected in these households? And as you would expect from the previous plot, we found no difference in the types of bacteria found in homes where children went on to be recurrent wheezers versus those who did not develop disease. When we looked at the children who developed atopy versus those that did not develop disease, this is a p-value. This is our significance cutoff. And anything that's above that is significant. If it's in this quadrant, it is a bacteria that is enriched in the households of atopic children. If it is in this quadrant, these are bacteria that are enriched in the homes where the children did not develop disease. And you can start to see a few organisms now begin to emerge when we make this comparison. This becomes even more pronounced when we compare the children who develop both atopy and recurrent wheeze compared to those who don't. So what it looks like is that it's a lack of exposure to organisms in the environment that is associated with disease development. And that fits with the hygiene hypothesis. And this is in the inner city, and this is the environment where people said this bucks the trend for the hygiene hypothesis. It's a dirty environment, there's lots of microbial exposure, so the hygiene hypothesis doesn't apply here. But it turns out that not all microbial exposure is equal in the inner city environment, just as in any other environment. And in those households where children are exposed to this small number of bacterial taxa, they do not develop disease. So who are those organisms? Well, if you look at these ones here, they're a pretty interesting group of organisms. Bifidobacteria, which we know are essential commensals in the early infant gut. Prevotella, Lactobacillus. This includes Clostridial species, which are known modulators of immune response, and several others. If we look at these here, again, we see some of the same species. I'm sorry, this has come up a little too soon. So we've got 82 bacterial families of interest. And what was striking to us is these have all been described in gut microbiomes. So it again fits with our hypothesis that its exposure to these microbes in the environment may be altering the gut microbiome composition in a manner that protects against disease development. And so I told you that we'd come back to Fido. We know that in early life, exposure to dogs, and to a lesser extent cats, protects against allergic disease development in childhood. And we wondered, maybe this is also microbially driven. Maybe pets introduce organisms from the external environment into the household that are then picked up by the developing infant to populate their microbiomes, particularly that one in the gut. And so we set out, and this study was actually, it predates the other inner city study, we set out to ask, what's the bacterial community composition of homes in which there are dogs present, cats present, or no pets present, which we consider the bacterial desert. There just aren't a whole lot of things there. And this was what we found. Again, this is looking at those same three metrics to really summarize the bacterial communities that are present in these households. In each one of these plots, the middle is the no-pet samples that have had their bacterial microbiome profiled. Here are the dog samples, here are the cat samples. This is the same across each of the plots. And what you can see, much as we'd seen in the inner city, in homes that have no pets and would be considered a risk factor for developing disease, they have significantly fewer types of organisms present, they are less diverse, and they are less even. So again, very consistent with the previous findings in that the no-pet environment, which confers a risk for developing allergic asthma, is deficient or devoid of microbes. And again, when we looked at the specific organisms that were significantly enriched in the dog-owning homes, again, these are all species that we have previously identified, or not just us, other groups have previously described in the human gut microbiome. So then we really wanted to try and prove that the environment in the household actually influences the gut microbiome in a manner that may confer protection of the airways against allergic challenge. And that's a very difficult study to do in humans. Our IRB won't allow us to feed infants house dust. Apparently you're not allowed to do that. And so we turned to our favorite model. We turned to mice. And so we collected house dust from a home with 2 dogs present, and house dust from a home with no pets present. And we used a model that has been previously described in the literature. And it involves feeding the house dust, the different house dusts, to mice, gavaging them daily for 7 days. We normalized the weight of dust that we get to these animals. We fed them for 7 days. And then over the next 3 weeks, we continued to supplement them twice per week with this dust to try and keep up that microbial exposure. And during this 3-week period, we challenged the airways of these mice intratracheally with cockroach allergen. And so this is a standard model that's been out there that allows you to examine allergic responses in the airways to cockroach allergen challenge. And what we found was, to us, expected but quite astounding that it was so profound. When we looked at the airways of these mice, an allergic response is characterized by Th2 responses, and the cytokines that are characteristic of that response are IL-4 and IL-13, amongst others. And what we could see is that the animals who received the dog-associated house dust, shown here in the hatched bars, had significantly reduced expression of these cytokines associated with allergic response compared to either the no-pet-fed animals or the control animals who we did not manipulate in any way. Another characteristic of allergic response is GOB5 hyperexpression. This is the gene that encodes for mucin and controls mucin hypersecretion. And again, what we could see is that the animals who received the dog-associated house dust had significantly reduced GOB5 expression in their airways. And this was really beautifully supported by histological slides of the airways of these animals. These blank spaces here are the air spaces in the lungs of the animals. These are control animals. We didn't manipulate them in any way. We didn't expose them to cockroach allergen. So they have pristine airways. These are the animals who we did not supplement, but we simply exposed them to cockroach allergen. And here you can see that the airspace is entirely occluded with this pink-stained mucin, and that these airspaces are entirely surrounded by a swell of inflammatory response cells that are presumably involved in expressing these pro-inflammatory cytokines, the Th2 cytokines that we see here. In the no-pet supplemented mice, we see some airspaces remaining, but there's a large amount of occlusion, a large amount of mucin hypersecretion, and again, the immune response is pretty significant around those airspaces. In contrast, the dog dust-fed animals have airspaces that are as pristine as the control animals, showing us and supporting this assertion that we are seeing a muting or a downregulation of the pro-inflammatory response that is characteristic of the airway allergic response. But remember that the hypothesis was that this would occur based on changes in the gut microbiome. So again, we went back to profiling the bacterial types that are present in the ceca of our mice that were our control animals and that were not protected, our animals that received the no-pet associated house dust, and those that received the dog-associated house dust. And remember I told you that the distance tells you how similar the communities are? You can see that the controls are out here by themselves as a group and are quite distant from the other two groups. So they have a very distinct bacterial community in their ceca. The dog-fed, dog dust supplemented animals and the no-pet supplemented animals are more similar, but these are still significantly different. These still cluster into one group which is distinct from this group. So there's differences in the gut microbiome of these animals that are either protected or unprotected against allergen challenge. And the next question was, who are the organisms associated with protection? And so this is where we took the profiles of organisms in the protected and the unprotected animals and simply compared them. Which organisms are enriched in the unprotected animals and which are enriched in the protected animals? And to go through this, every bar is a taxon, or a group of organisms, a family of organisms that is enriched. The color of the bar tells you whether they are enriched in the protected animals in red are enriched in unprotected animals in blue. This beautiful tree, phylogenetic tree here, tells you which phylum those organisms are related to. And you can see from this that the majority of organisms that are most significantly enriched in the protected animals belong to the firmicutes, this one phylum that we know is a key phylum in the human gut microbiome as well as in other mammalian systems. The height of these bars tells you just how much the magnitude of change is across these two groups. So we were really interested in these four groups here, which represent the organisms that were most significantly enriched in protected animals. And we were particularly interested in this one that cropped up, Lactobacilliaceae, and the name of the organism that's represented in that taxon is Lactobacillus johnsonii. And why we got very excited about this organism is that it becomes highly dominant in the maternal vaginal tract just before the birthing process. So we thought this is possibly an organism that really directs the show with respect to who co-colonizes in that developing gut microbiome in very early life. So for us, it was one of the primary targets or primary organisms that could potentially change the gut microbiome and alter its composition and function in a manner that protected the airways. I should also say Lactobacillus species are largely oxytrophic. They need partners. They are not able to produce all of the amino acids that are essential for their longevity. And so the fact that these organisms are oxytrophic tells us that it has to change the composition of the community to promote specific organisms that promote the enrichment of and the longevity of the Lactobacillus species. So we thought for a number of reasons this was a really good target organism to go after. So we took another set of mice, we fed the poor creatures more dog-associated house dust, and then we took their ceca and isolated on Lactobacillus selective media the dominant Lactobacillus species in the gut of those mice. We looked at about 20 different colonies across 4 different mice, and every single one of them was Lactobacillus johnsonii. So our molecular profiling had accurately told us who the dominant Lactobacillus was in these protected animals. We batch cultured, so we made large-scale cultures of this one specific organism. And then we went back and repeated the same experiment again in the Murine model of cockroach allergen challenge. But this time, we simply fed the single organism about 1 by 10 to the 7 colony-forming units per supplement of this organism. We continued the supplementation in the test animals twice per week during this period when we were challenging the airways of these animals. And again, we were very excited by what we found. First thing up here is one way we can measure how allergic the response is in the airways or how responsive the airways are is by challenging airways with a compound called methacholine. And so this gives you a measure of how responsive the airways are. The more responsive, the more allergic. And what we could see is that in the Lactobacillus johnsonii fed animals, we significantly reduced bronchial hyperreactivity in these animals. Much as like we had seen with the dust experiments, in the airways of these animals, we now see with just Lactobacillus johnsonii supplementation a significant reduction in IL-4, IL-5, and IL-13. Again, the hallmark characteristic cytokines or responses of the immune cells that mount the allergic response. We've aggregated these both at the expression level, the mRNA level, and at the protein level. And again, if we look at the airways, these are the animals who were just challenged with cockroach allergen. Again, their air spaces are full of pink staining mucin, and they have inflammatory cell infiltrates around the air spaces. These are the animals who received the Lactobacillus species. And quite interesting, they are protected, clearly at a molecular level and grossly with respect to their histology. But I would say that they're not quite as protected or as pristine as we saw with the dust supplemented animals. We see a little bit of mucin secretion at the airway surface, but for all intents and purposes, these are protected animals, and the majority of this protection appears to be coming from supplementation with Lactobacillus johnsonii. So then we began to think, This is Allergen. I should say at this stage that we repeated this with a distinct allergen challenge with ovalbumin, and we saw exactly the same response, that we could quench the allergic response to either of these allergic challenges to the airways. We began to wonder if this was more of a universal protective phenomenon that we had created in these animals, and whether we needed a live organism to confer this protective effect. So we set about another suite of experiments where we supplemented the animals, this time in a model of respiratory syncytial virus infection. We chose that viral infection because children who have a severe response to RSV in the first 2 years of life that requires hospitalization have a significantly increased risk of developing allergic asthma. It's seen as an allergic asthma, kind of an asthmagenic viral infection. And so this time when we supplemented the animals, we supplemented them with heat-killed Lactobacillus johnsonii and equal numbers of the live organism. And so what we see, again, the airway responsiveness is only significantly reduced in the animals who receive the live supplement of this organism. Again, in the airways, and this is a little difficult to see, I would concur, that the airways are significantly protected and have significantly less mucin hypersecretion in their airways, only in the live organism supplemented mice. In the airways, we took a look at the Th2 allergic response characteristic cytokines, and much as we'd seen with the challenge with allergy, when we challenge with respiratory syncytial virus, we see a significant downregulation of all of these cytokines, the Th2 cytokines. We also saw a downregulation of a distinct cytokine associated with another subcellular group of adaptive immune response, which is quite interesting that we are downregulating several aspects of the adaptive immune response. What we didn't see was a significant abrogation in the interferon gamma response. This response is mounted to allow the host to clear the viral infection. So this is a good thing, that we're not seeing a significant reduction in this. It suggests to us that the organism, the mouse, still has the capacity to clear the viral infection, but does not mount the Th2 and Th17 response in the airways that is what causes the pathology associated with the viral infection. We also looked at the mesenteric lymph nodes of these mice. So these are the lymph nodes that are associated with the gastrointestinal tract, to ask whether the cellular populations of the immune response in this site are altered. And we found that they were. Dendritic cells are antigen-presenting cells that take allergen or foreign compounds and present them to T cells in the mesenteric lymph nodes. And that, with other signals, drives T cell proliferation. And that's what drives the proliferation of these adaptive immune responses that we see in the airways of these animals. And what we found was that the dendritic cells were significantly reduced in numbers, they were significantly less activated, and there were significantly fewer T cells associated with the mesenteric lymph nodes of these animals, suggesting that the changes in the microbiome and the activities of the microbiome in the gastrointestinal tract cause changes in the priming of key antigen-presenting cells in the lymph nodes associated with the GI tract. So truly suggesting that the protective effect we're seeing is potentially mediated by changes that occur in the mesenteric lymph nodes close to the gastrointestinal tract. So then we asked, does Lactobacillus johnsonii actually change the gut microbiome? Is this just because we've added in a large amount of this organism and it's driving the show? Or does it cause, as we suspected, changes in the composition and the function of the gut microbiome? Again, this is just another way of looking at microbiome profiles. Again, proximity tells you similarity in the communities that are present. Each one of these dots represents a cecal microbiome profile of control animals, cockroach allergen challenged animals, Lactobacillus johnsonii supplemented animals, and Lactobacillus johnsonii cockroach allergen challenged animals. And what you can see is that the Lactobacillus johnsonii significantly changes the composition of the gut microbiome in these animals. And that even though cockroach allergen challenge seems to change this composition, it never becomes like that of the control animals challenged with cockroach allergen. So suggesting that this organism can induce a very dramatic shift in microbial community composition. What was striking to us is, remember those other 3 organisms that I told you were significantly enriched in protected animals that we fed dog-associated house dust? They were not enriched here. And what that suggests to us is that these are environmentally sourced organisms that work in unison with the Lactobacillus species to elicit full protective effect in the airways. And this is something that we are pursuing right now, that these organisms as a consortium maybe provide full protection of the airways when fed into the gut and cause changes in the gut microbiome composition and function. So we conclude that the local environment exposures are related to clinical outcomes of childhood allergic disease, and specifically the microbial exposures in the home are related to whether children develop disease or not. The gut microbiome, we've also shown, can actually be influenced by the environmental exposures in the home, by house dust. We suspect that this is largely due to the microbes there, but we're not discounting the fact that there may be other allergenic components in the house dust that maybe drive this compositional shift in the gut microbiome also. And finally, there's been several studies that have shown if you ablate the gut microbiome with antibiotics, you can enhance the pathogenic effect of an infective agent like influenza A or several other infectious agents. And that's wonderful. It proves that the gut microbiome modulates response to infection. But I think what's really exciting is that we've been able to protect the host against viral infection and allergen challenge, a range of very distinct pathological agents. And this leads us to be very hopeful that we can really translate this into truly effective therapies that target the gut microbiome as a therapeutic target. So what's next? Well, we view chronic inflammatory diseases, not just allergic asthma, but a variety of other diseases that we study, like inflammatory bowel disease and immune activation in HIV infected patients, as the veritable burning forest. And what we want to do is to restore it to its original state, or what it should look like. And we think that one of the ways to do this is through microbial supplementation. And I say that because I do believe that that's a better approach rather than feeding in a single metabolite, for example. I think these organisms produce multiple functions. They also control the stoichiometry of the system. They have the enzymes that control how much product is produced. So they maintain those physiologically relevant concentrations of the compounds and the metabolites that are necessary to maintain homeostasis in the system. And we need those checks and balances in place, for me, I think, to maintain that state of immune homeostasis. And so what we have at the moment are kind of 2 extremes. We have over-the-counter and prescribed probiotic formulas. These are largely consistent of 1 or 2 or a handful of species. They typically are organisms that provide very similar functions. A large amount of them are fermentative, and they can produce, for example, short-chain fatty acids, but they don't confer the full suite of functions that are necessary from a healthy microbiome to confer protection against infection or allergic challenge. And while they may work for some people, that may be because the ancillary actors, the supporting actors that these organisms need to survive in that ecosystem in the gut are present. But remember that in chronic inflammatory disease, one of the characteristic hallmarks is a significant depletion of bacterial diversity. So it's very likely that you have a variety of background organisms across a variety of patients, and it's by chance that you, with probiotic supplementation, will have the appropriate supporting actors to confer a beneficial effect of this kind of supplementation on the host. The other approach we have is kind of the other extreme, fecal transplantation. It has been incredibly efficacious for one specific indication, Clostridium difficile infection, that is recalcitrant to antimicrobial therapy. There has been lots of conversation about it being applied for pretty much every other disease under the sun that we have associated with the microbiome dysbiosis. But I would really be very cautious as we move forward with this. There is huge variation in the types of communities we are transferring to the patients who are receiving them. They're across a broad swath of ages, genders, ethnicities. We also have no idea what's the perfect match. Remember that the gut microbiome develops very dramatically in the first year of life and continues to develop at a slower pace afterwards. We don't know what that means for physiological or immunological development. We haven't done the studies yet. What we know on the other end of the spectrum is as people age, their microbiome essentially disassembles, and that occurs in parallel with immune senescence. So what are we really doing when we transfer a 56-year-old woman's fecal material to a 3-year-old child with inflammatory bowel disease? While we may have short-term, really very impressive outcomes, what's the long-term implication of that transfer? We don't have any idea. There's a lot of work to be done, but I would urge caution. I think the more strategic approach is a rational design of organisms, the small group of organisms necessary to provide a consistent suite of metabolites that we know are associated with health and that we know confer anti-inflammatory attributes if that's what we're trying to do, if that's what we're trying to treat. And finally, I would say that we need to know far more about the system. There is several careers worth of research to be done in this field. I think we've found the black box. I think we're just at the very, very beginning of understanding and leveraging these microbes that we have evolved with over eons of time to promote human health. But I would urge caution in how we move forward with this and really base our therapeutics on real science and really understanding what these microbial transplants or supplementations can actually do for individuals. And finally, I'd like to acknowledge a wonderful group, my ecosystem, my research ecosystem, which is highly diverse and multifunctional and runs the gamut from physicians to microbial ecologists, old school microbiologists, and a range of other skill sets. I particularly want to point out Homer Boucher and Kei Fujimura, who've been long-term collaborators, and much of the work that I presented today is due to their efforts. Also to Chris Johnson and Ed Zerotti at Henry Ford Hospital, who provide us with amazing birth cohorts to work with, as have Jim Gurn and Bill Bussey. Owen Brody at Lawrence Berkeley National Lab, who introduced me to this field and provided me with the tools to get started in profiling these communities, as well as Nick Lukacs and Tina DeMoore at University of Michigan, who performed much of the mouse studies that I described in this presentation. And finally, but not least, our funders from the NIAID, NHLBI, NCAM, and a variety of foundations and pharmaceutical collaborators. And I'm very happy to take any questions. Thank you.
Thank you for a terrific talk. Before we move to questions, I just want to say a couple points. First, there will be a reception outside afterwards, so we'll keep the questions a little bit short so that people can have a chance to talk to Dr. Lynch personally. Second, if you do ask questions, which we strongly encourage, please step to a microphone. We're recording this, and if you ask a question and there's no mic, then it doesn't end up being recorded, so we don't really hear your question as much. So please use the mics. We'll have roving mics as well. And third, I'd like to thank a number of people who are just essential for making this show happen. First, the funders, Herman and Bea Silverstein. We could not do these kinds of lectures without their funding. Second, the staff of the Center for Genetic Medicine, in particular Michelle Money, who did a lot of work to put this together, and Northwestern University for housing this whole operation. So let's move to questions, and I open the floor.
Are there parallel studies from children in rural communities or in countries where the populations are largely agrarian to compare with?
Yes, there's a really beautiful paper from Jeff Gordon's group who compared infants and children as well as adults across 3 geographically and ethnically very diverse communities in the US, an Amerindian tribe, and also in Malawi. And they have very distinct community compositions. And it was largely driven, was the conclusion, by diet, in that those more in the developing nations had a lot more plant-based polysaccharide consumption, whereas in the Western nation, in the US, there was a lot more simple sugar consumption that was associated with the differences in microbiome across those 3 groups. That's probably the biggest study that has been done to date. But yes, they dramatically differ based on geography and ethnicity, which encompasses diet. Yeah.
Thank you, that was brilliant. A question regarding your analogy to the forest fire and the reforestation. If you throw seeds into a burning fire, they're going to burn up. You have to wait until it's gone and the fire's gone. In many animal models of chronic inflammatory diseases, and even in your asthma model, you can prevent the disease by the exposure. But once you've got the disease, it's difficult to affect it by repopulating with bacteria. And one of the concerns is C. difficile is pretty easy. It's one organism,
I agree.
and you can repopulate and prevent it. But in disorders like inflammatory bowel disease, once you've got it burning, one questions whether repopulating is actually going to affect it until you sublimate the fire.
Yeah, I absolutely agree, and that's why I think there have been some studies in inflammatory bowel disease with fecal transfer that have not been highly successful. I think they've had a 30% or so success rate. I think we shouldn't think about, just going with one therapy. We shouldn't throw the baby out with the bathwater. We have a number of effective therapies to quench immune response in inflammatory disease patients, for example. I think that is the opportunity then to come in with a reconstitution approach, that it's not just simply you throw on an inoculum into the burning flames, as you say, that will not work. We need to manipulate the host immune response, so we now generate an ecosystem that is receptive to that inoculum that we're trying to engraft into those individuals. And I think that that's going to be a multi-pronged approach, not just simply recolonization, but using the other components of our armory that we have. One of the other things that we've been really interested in is that microbes naturally compete with one another as well. And so thinking in terms of who are the dominant organisms that are driving the dysfunction and immune dysfunction that we see in those patient populations, and can we leverage the natural competition between microbes to oust the pathogenic organism? It's another approach that we've been considering, that it's not going to be a one consortium fits all. We need to understand what are we trying to treat and who are the organisms that we're trying to displace in a pathogenic scenario, and then who are the best group of organisms to outcompete that organism and restore the community. So I think we can use a number of tools, and it will not simply just be about recolonization or rehabilitation of the microbiome with one group of organisms.
Question here. So how, given the complexity of post-microbial interaction and even the microbial competition, how feasible it is in the near future, within the next 10 years, for example, you sequence your entire genome and then you sequence your gut flora, and then derive the equation that you should transplant this and that combination, so that I know it's an unanswerable question. But one comment is, you must have noticed that in Chinese medicine, traditional medicine, that has been practiced for maybe over 1,000 years by only a small group of practitioners of the fecal transplantation. There, the emphasis was also on like childhood or baby fecal material to be transferred. So I wonder if you have comments on that.
Yeah, I do. Yeah. When you say about Chinese medicine, I remember when we first got this data showing that the gut microbiome modulation could affect the airways. I was so excited, and a very good friend and colleague of mine, Wanlin Song, who's Chinese, was in my office, and I showed him the data. I was like, this is fantastic. And he said, We've known this in Chinese medicine for decades, Sue. What are you talking about? When you've got an airway problem, it is the fire from the belly that is burning the lungs. So, I feel like we go 3 steps forward, 2 steps back. I'm also an optimist, and I do believe that we will get to a point where we can personalize your therapeutic based on your community. I think the evidence for that, it comes from, for example, Peter Turnbaugh has been showing that your capacity to transform a drug called digoxin is dependent on whether a specific organism is present in the gut microbiome or not, and that dictates its bioavailability. I do believe that we will get to that point. I don't know if it's in the next 10 years, but I think our feeling is that if we want to rebuild communities, we need to understand the blueprint for building them in the first place and understanding what goes awry when disease develops in childhood. And that's why we focus on the early life gut microbiome. I think a lot of the answers are there because we're looking at the development of the microbiome, not just compositionally, but functionally also. Which are the consistent functions that are necessary to promote a healthy microbiome? And I think from there, we can extrapolate groups of organisms, for example, that may be useful for certain groups of patients. The other thing I think we can leverage with the microbiome is improved patient stratification. So we know in asthma and IBD, there's a number of different endotypes that we see. Anyone who treats the patients with those diseases knows that there's groups of patients with different manifestations of disease. And so one of the things we've considered is that maybe be driven by a different type of microbiome. And we in fact have evidence for this in inflammatory bowel disease, in HIV patients with pneumonia, and in chronic sinusitis. So this seems like it's a very consistent signal that we can see that specific immune phenotypes of patients are associated with a very specific dysbiotic community. What we've largely done before this is we've lumped everybody together. Everybody's got IBD. And we've not thought about refining based on disease endotypes, for example, or severity of disease, which kinds of microbial communities maybe drive those specific phenotypes. So I think there's a lot to leverage from the microbiome, and I am still optimistic that in 10 years' time, I can come back to you and say, look, we have more personalized medicine than we had 10 years ago, and that there has been some movement in the field based on the findings of this emerging field of research.
And I think on that very positive note that we will thank Dr. Lynch and move to the reception.